Hey everyone, and welcome back for another deep dive. Today we're going to be unraveling the world of inheritance.
Sounds
It
fascinating.
is. You guys sent over a ton of excerpts, and there's some seriously cool stuff in there.
Like what?
Well, we've got everything from the history of genetic research, all the way back to Mendel and his peas.
Of course.
And we've got key concepts like genes, alleles, inheritance patterns, and even stuff like sex determination and genetic disorders.
Wow, we've got a lot to cover.
We do, but I think it'll be fun. It's amazing to think that even way before we had, formal genetics, humans were already noticing those inheritance patterns.
Oh, yeah, totally. Think about those early farmers who were selectively breeding their crops and livestock. They were basically doing practical genetics, even without all the fancy terms we use today.
It's true. They were really laying the groundwork for what would become a whole scientific revolution.
Right.
But I gotta say, the real game changer was Gregor Mendel and his work with pea plants. I mean, he's considered the father of genetics for a reason.
Absolutely. Mendel's approach was totally groundbreaking. He didn't just observe things, he quantified, analyzed, and he applied hardcore math to biology.
Which was unheard of at the time.
Exactly. And through his meticulous work with pea plants, carefully studying those 7 contrasting traits like plant height and seed shape, he uncovered these fundamental principles of inheritance that still form the bedrock of modern genetics.
It's incredible how he took something as seemingly simple as peas and unlocked these profound insights into heredity.
It is.
Can you walk us through some of those key discoveries? I remember learning about Punnett squares back in high school, but I'm ready for a way deeper dive now.
Of course. Let's start with the foundation genes and alleles. Mendel's work showed that traits are passed down through these units of inheritance called genes, and each gene can have different versions, which we call alleles.
Okay, so like in the case of pea plant height, there's an allele for tall and an allele for dwarf.
Exactly.
So it's almost like a recipe where you can substitute different ingredients and get slightly different results?
Yeah, that's a great analogy. What's even more fascinating is that Mendel discovered some alleles are dominant, meaning their effect will always be expressed even if an organism only carries one copy of that allele. Like in pea plants, the tall allele is dominant over the dwarf allele. So even if a plant inherits one tall allele and one dwarf allele, it will still grow tall.
Right, that explains why in that first generation of his hybrids, all the plants were tall, even though they had one dwarf parent.
Exactly.
But then things got interesting in the second generation, right?
Exactly. When Mendel self-pollinated those tall hybrids, the dwarf trait reappeared in the next generation, and it did so in a specific ratio, 3 tall plants for every 1 dwarf plant.
Wow.
And this observation led him to formulate the principle of segregation, which basically states that during the formation of sperm and egg cells, those paired alleles, they separate from each other so that each gamete only carries one allele for each trait.
So it's like a genetic coin flip determining which allele gets passed down.
Yeah, you could say that.
And that random assortment is what contributes to all the incredible diversity we see in nature.
Precisely.
Awesome. And those Punnett squares you mentioned, those are a visual tool that helps us map out all those possible combinations of alleles
Right.
and then predict the probability of different genotypes and phenotypes in the offspring.
Exactly.
Right. Genotype being the actual genes an organism carries, and phenotype being how those genes actually express themselves.
Yes.
Like those tall or dwarf pea plants.
Right.
But Mendel also experimented with multiple traits at once, right? I vaguely remember that making things even more complex.
You are remembering correctly. Mendel's experiments went beyond single traits. He investigated how 2 traits are inherited simultaneously, like seed color, yellow versus green, and seed shape, round versus wrinkled. And what he found was that these traits were inherited independently of each other.
Okay, so inheriting, like yellow seeds didn't automatically mean you'd also inherit round seeds. It was separate coin flips, genetically speaking.
Yeah, exactly. This is his law of independent assortment, another cornerstone of genetics.
So you could have all sorts of combinations, round and yellow, wrinkled and green, you name it.
Precisely.
It's amazing how these seemingly simple principles can lead to such incredible variation.
It is. It is.
But I've always wondered, why was Mendel's work initially ignored for so long? It seems like such a groundbreaking discovery, but it took decades for the scientific community to really catch on.
Yeah, it is a bit of a curious case in the history of science. Mendel published his findings in 1865, but it seems his work was simply ahead of its time.
Like people weren't ready for it.
Right. The scientific community wasn't quite ready to grasp the significance of his discoveries. It wasn't until around the turn of the 20th century, with the advent of better microscopes and the discovery of chromosomes, those thread-like structures within the cell
Oh,
nucleus.
so that's when people started to really appreciate Mendel's work.
Exactly, yeah. And what's really remarkable is that when they observed how those chromosomes behave during cell division, the way they segregate and assort it, perfectly mirrored Mendel's principles of inheritance. So suddenly, those abstract concepts of genes and alleles had a tangible physical counterpart in those chromosomes.
Like a missing piece of the puzzle had fallen into place.
Exactly. And it was pivotal. It united those different strands of research and really solidified the foundation of modern genetics.
So it really led to that chromosomal theory of inheritance,
Exactly.
which basically states that genes reside on chromosomes and their behavior during cell division explains those patterns of inheritance that Mendel had so meticulously observed.
Right.
It's like suddenly seeing the clockwork behind the face of a clock.
Yeah.
It all makes sense now.
Yeah, it does.
But other scientists built upon that foundation, right? Taking those basic principles and revealing even more complexity in the world of inheritance.
Absolutely.
Like Thomas Hunt Morgan and his work with fruit flies.
Yeah. You're absolutely right. Morgan's research with those tiny fruit flies, Drosophila, added another layer to our understanding of inheritance, particularly with his concept of linkage, where he observed that genes located on the same chromosome, they tend to be inherited together.
So it's not always completely random assortment.
Right.
Some genes are like partners in crime, always sticking together.
That's a good way to put it. Genes on the same chromosome are physically linked and therefore more likely to be passed down as a package deal. But even here, there's a twist. Morgan also discovered that this linkage, it isn't absolute. Sometimes those linked genes can be separated during cell division through a process called recombination.
So even those partners in crime can have a falling out. What causes that?
Recombination happens when chromosomes swap segments of DNA during a special type of cell division called meiosis. This is the process that produces sperm and egg cells. And this shuffling of genetic material, well, it leads to new combinations of alleles. It just further amplifies the genetic diversity that we see in nature.
So it's like those gene partners are swapping outfits, creating new looks, and adding even more variety to the mix. But how did scientists even figure out what was going on at such a microscopic level? Were they just looking at these flies under a microscope and making these incredible deductions?
Well, that's where the power of scientific inquiry really shines. By meticulously tracking the frequency of those recombination events, Morgan and his team were able to actually map the location of genes on chromosomes. And this laid the foundation for genetic mapping. It's a powerful tool that's still used today to locate genes on chromosomes and understand the organization of genomes.
Okay, so we've come a long way from Mendel's peas. It's incredible to think how those foundational principles have led to such a complex and sophisticated understanding of inheritance. But even with all these discoveries, I have a feeling there are even more layers to this intricate tapestry of genetics.
You're right, there are. Mendel's work definitely set the stage, but inheritance is even more intricate than initially thought. There are so many fascinating nuances and variations that we haven't even touched on yet.
Well, I'm ready to delve deeper. Let's take a closer look at some of those complexities and see what other secrets we can unlock.
I'm happy to guide you. It's remarkable how Mendel, without even knowing about chromosomes, essentially predicted their behavior.
Yeah, it's like he was a genetic visionary. But even with those insights from Mendel and what Morgan discovered about linkage and recombination, it turns out inheritance is even more intricate, right?
Absolutely. Not all traits follow those simple Mendelian patterns of dominance and recessiveness. There's a whole spectrum of inheritance patterns that add layers of complexity to this genetic tapestry.
So it's not always as straightforward as those tall versus dwarf pea plants.
Right.
There are other factors at play.
Exactly. Let's take incomplete dominance, for example.
Okay.
In this case, neither allele is completely dominant over the other. Instead, they blend together in the heterozygote, resulting in an intermediate phenotype.
Ooh, I love a good blend. Got any examples? You know me, I'm a visual person, so concrete examples always help me wrap my head around these concepts.
Sure. Imagine those vibrant snapdragon flowers.
Okay, yeah.
If you cross a true breeding red snapdragon with a true breeding white one, guess what color you get in the F1 generation?
I'm going to guess pink.
You got it.
A beautiful fusion of
Yeah.
the two parent colors.
Exactly. You get those pink snapdragons because neither the red nor the white allele is fully dominant. It's a stunning example of how genes can interact to create a whole spectrum of variations rather than just distinct categories.
Pink snapdragons, nature's perfect compromise.
You could say that.
Okay, what about codominance? What's happening there? Is it similar to incomplete dominance?
Well, in codominance, both alleles are expressed equally in the heterozygote.
Okay.
Think of it as a power-sharing agreement where both alleles get to have their say.
Ooh, I'm intrigued. Tell me more.
Okay, well, a classic example is the ABO blood group system in humans.
Oh, yeah. Okay.
There are 3 alleles involved, IA, IB, and I. IA and IB are co-dominant, meaning if you inherit both of them, you'll express both A and B antigens on your red blood cells, resulting in the AB blood type.
So it's not a blend like the pink snapdragons.
Right.
Both traits are on full display. Each allele makes its own unique contribution.
Exactly. And then you have the I allele, which is recessive to both IA and IB. So if you inherit 2 copies of I, you'll have O blood type. This means your red blood cells won't have A or B antigens.
Ah, that explains why O is the universal donor.
Yeah.
Because it doesn't have those A or B antigens that could trigger an immune reaction in people with other blood types. This is all making so much sense now.
It is fascinating how these basic genetic principles can shed light on real-world things like blood transfusions.
For sure. And there's even more to it, right? As we dig deeper into inheritance, we start to see traits that are influenced by multiple genes working together, not just one.
Right, we call that polygenic inheritance.
It's polygenic. Now that sounds complex. What kind of traits are we talking about here? Are these the traits that have a wider range of variation?
You got it. Think about traits like human height or skin color. These traits don't fall into neat categories like tall or short, light or dark. They exist on a continuous spectrum with subtle gradations.
So instead of just one gene for height, there might be dozens of genes, each contributing a little bit to your final height. Like a team of artists, each adding their own brushstroke to create this masterpiece of complexity.
I like that analogy. And to add another layer of complexity, environmental factors can also influence those polygenic traits.
Oh, right. So that's why even identical twins who share the same genes might end up with slightly different heights or skin tones, depending on things like nutrition or sun exposure. It's that whole nature versus nurture debate.
Exactly.
It's also intricate. But wait, isn't there also something called pleiotropy? I always have trouble saying that one. What's that all about?
Pleiotropy is this really fascinating phenomenon where a single gene influences multiple, seemingly unrelated traits. It's a reminder that our bodies are these complex systems with intricate feedback loops.
Okay, my mind is officially blown. Got an example of this genetic multitasking.
Sure, think about the disease phenylketonuria, or PKU. It's caused by a mutation in a single gene that affects the way the body processes the amino acid phenylalanine. And this single mutation can lead to a range of symptoms from intellectual disabilities to reduced pigmentation in the hair and skin.
So one tiny change in a single gene can have ripple effects throughout the whole body.
Exactly. It highlights the interconnectedness of our genes and how even small changes can have a huge impact on our development and health. It really speaks to the complexity of the human body.
This is all so fascinating, but I have to admit, my head's spinning a little bit. It's like peeling back the layers of an onion. We just keep revealing more and more intricate details.
I understand. But it's through understanding these complexities that we can really appreciate the amazing diversity of life. And it helps us unravel the mysteries of genetic disorders and figure out new ways to diagnose and treat them.
Speaking of mysteries, what about one of the biggest mysteries of all? How is sex determined? How does genetics determine whether we become male or female? I remember learning about the X and Y chromosomes, but I'm ready for a deeper dive.
Well, as with a lot of things in genetics, the answer lies in our chromosomes. Remember, humans have 23 pairs of chromosomes, 22 pairs of autosomes, which are the same in males and females, and 1 pair of sex chromosomes.
Okay, so those sex chromosomes are where the magic happens.
Exactly.
They hold the key to determining our biological sex.
Right. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome.
So it all comes down to that Y chromosome. It's the defining factor for being male. If you get a Y chromosome, you develop as male.
That's generally how it works in humans, but as with most things in genetics, there are exceptions.
Of course.
There are organisms out there that have completely different systems for determining sex.
Right, right.
But sticking with the human system, during sperm formation, those sex chromosomes segregate, just like Mendel observed with his pea plants. So half the sperm will carry an X chromosome and half will carry a Y chromosome.
And when that sperm meets the egg, which always carries an X chromosome, it's like a 50-50 chance of whether you'll end up XX female or XY male, like a genetic coin flip.
You got it. It's a random event that determines our biological sex. And I think it's really important to emphasize that this process is totally random, you know? We need to debunk those outdated beliefs that might blame mothers for the sex of their children.
Absolutely. We need to challenge those misconceptions.
Uh-huh.
And speaking of challenges, we can't ignore those times when genes don't quite behave as expected. That's where genetic disorders come into play, right? When those mutations we talked about earlier disrupt these intricate processes.
You're right. Sometimes there are changes in our DNA sequence, those mutations that can lead to genetic disorders.
So those mutations can arise spontaneously or can be caused by things in the environment, like radiation
Right.
or exposure to certain chemicals. And they can range from single letter changes in the DNA to these like larger alterations in the chromosome structure.
Right. They can be pretty small or pretty big.
So our DNA is under constant threat. It's like a genetic battleground, with our cells fighting to maintain the integrity of that genetic code.
In a way, yes. But our cells actually have remarkable repair mechanisms.
Oh, that's good.
Yeah, they're constantly at work fixing those errors in our DNA. It's like they're these dedicated proofreaders tirelessly scanning for typos and making corrections.
Okay, that's a relief.
Yeah, but sometimes those repairs, they're not perfect.
Wow.
Or the damage is just too much. And that's when mutations can slip through and potentially lead to problems.
So let's talk about those genetic disorders. What are some of the key things we need to know about how these mutations mess things up?
Well, generally speaking, genetic disorders can be classified into two main categories: Mendelian disorders and chromosomal disorders.
Okay.
Mendelian disorders, named after our friend Gregor Mendel, are caused by mutations in a single gene.
So like a single typo in the code that can completely change the instructions.
Yeah, that's a great analogy. Just like a typo can change the meaning of a sentence, a mutation in a single gene can change the function of a protein, or even prevent it from being made at all. And that disrupts those essential biological processes.
And what about those chromosomal disorders?
Mm-hmm.
What's happening there? Are those caused by more extensive errors?
Yeah. Chromosomal disorders involve larger scale changes in the chromosomes, things like an extra copy of a chromosome or even a missing chromosome. These errors happen during cell division when those chromosomes are being replicated and distributed to those daughter cells.
Okay, so it's not just a typo. It's like a whole page is missing or duplicated in the instruction manual.
Yeah, a much bigger alteration
Why
that
not?
can really impact development.
So let's talk about some specific examples of these disorders. What are some of the most common Mendelian disorders?
Okay, sure.
I know we talked about PKU before.
Right, well, there's a whole range of them. Some other well-known examples are color blindness, hemophilia, sickle cell anemia, and of course, PKU.
Color blindness. That's where people have trouble distinguishing certain colors, right?
Yeah.
I remember learning that it's more common in men. Is there a genetic reason for that?
You're right, it is more common in men, and it's often caused by mutations in genes located on the X chromosome.
Oh, okay.
Remember, men only have one X chromosome, while women have two. So if a man inherits an X chromosome with a mutation in a color vision gene, well, he's going to express that trait.
He doesn't have backup.
Exactly. Women, on the other hand, have that second X chromosome.
Right.
So they're less likely to be affected unless they inherit two copies of that mutated gene, one from each parent.
Ah, that X chromosome strikes again. It seems to be involved in quite a few genetic disorders.
It does. It does.
What about hemophilia?
Yeah. Hemophilia is a bleeding disorder caused by mutations in the genes that code for proteins that are involved in blood clotting. Those proteins are really important, you know. They help stop bleeding after an injury. And just like colorblindness, hemophilia is also linked to the X chromosome and follows that sex-linked inheritance pattern.
So more common in males.
Exactly.
And sickle cell anemia. That sounds pretty serious. I know it affects red blood cells.
It is. It's a serious blood disorder that actually changes the shape of those red blood cells.
Okay.
Instead of being nice and round and flexible, the cells become, well, they have this sickle shape and they're rigid and sticky.
That sounds painful.
They can be, yeah. And these abnormally shaped cells can actually block blood flow in the small vessels, which can lead to pain, organ damage, and a whole host of other complications. And it's caused by a mutation in the gene that codes for hemoglobin, the protein that carries oxygen in our red blood cells.
So it's like those sickle-shaped cells are clogging up the system.
Yeah, that's a good way to think about it.
And then we have phenylketonuria, or PKU.
Yeah.
That's the one we talked about earlier, where a single gene mutation can cause a cascade of effects.
Yes, exactly. PKU is a metabolic disorder that prevents the body from properly breaking down that amino acid phenylalanine.
Right.
It's found in a lot of foods, and if it builds up in the body, it can be toxic to the brain, which can lead to intellectual disabilities, seizures, and other neurological problems.
Oh, wow.
But luckily, with early diagnosis and dietary restrictions, people with PKU can live healthy lives.
That's good to hear.
Mm-hmm.
So those are some examples of Mendelian disorders,
Yeah,
single gene mutations that can have all sorts of effects.
exactly.
What about those larger scale chromosomal disorders?
Yeah.
What are some examples of those and how do they happen?
Well, some well-known chromosomal disorders are Down syndrome, Klinefelter syndrome, and Turner syndrome.
Okay.
And these disorders arise from errors during cell division, leading to an unusual number of chromosomes in the cells.
Down syndrome, that's caused by having an extra copy of chromosome 21, right?
Yes, that's right.
And I know people with Down syndrome often have those characteristic facial features
Right.
and may experience developmental delays.
That's right. It's also called trisomy 21
Okay.
because individuals with Down syndrome have 3 copies of chromosome 21 instead of the usual 2. This extra chromosome really messes with the normal balance of gene expression,
Okay.
and this leads to that range of physical and developmental characteristics.
And Klinefelter syndrome, does that also involve an extra chromosome?
Yes, it does. Klinefelter syndrome affects males
Mm-hmm.
and is characterized by having an extra X chromosome. So instead of that typical XY chromosome pair, they have XXY.
Okay.
This can lead to things like infertility, low testosterone levels, and other health issues.
And then there's Turner syndrome.
Yeah,
That affects females, right?
it does, yes.
Does it also involve an extra chromosome?
Actually, Turner syndrome is caused by a missing or partially missing X chromosome. So females with Turner syndrome typically only have 1 X chromosome instead of the usual 2. This can lead to things like short stature, infertility, and a variety of other health concerns.
So all of these chromosomal disorders are caused by these pretty dramatic changes in the number of chromosomes.
They are.
It's amazing how resilient the human body is, even with these challenges.
It really is. And by understanding these disorders, both the Mendelian and the chromosomal ones, we can develop better ways to diagnose, treat, and support those affected.
This has been an amazing journey into the world of inheritance. We started with Mendel's peas, and we've made it all the way to those mind-boggling chromosomal disorders.
It's been quite the deep dive.
It has. It's amazing how much we've learned since Mendel's time. But how do scientists actually study inheritance patterns in humans? I mean, we can't exactly do controlled breeding experiments like with pea plants, all right?
You're right about that. Ethical considerations prevent us from doing those kinds of experiments with humans, but we have other tools to work with. One of the most valuable is pedigree analysis.
Pedigree analysis, huh? Sounds like we're tracing the lineage of a prized racehorse.
Well, in a way, we are. Pedigree analysis involves constructing family trees and then tracking the inheritance of specific traits across generations.
Okay.
It's like putting together a genetic puzzle, We're looking for those patterns and clues to understand how traits are passed down.
So connecting the dots between grandparents, parents, children, and so on, looking for those recurring patterns.
Exactly. And by carefully analyzing those family trees, we can figure out if a trait is dominant or recessive, autosomal or sex-linked, and even estimate the probability of future generations inheriting certain traits. It's a really powerful tool.
Wow, that sounds like a lot of work. Like being a genetic detective.
It definitely requires a lot of attention to detail and strong analytical skills. And you never know, sometimes those pedigrees can reveal some surprising connections and even family secrets.
Ooh, juicy. So pedigree analysis is like looking into our genetic past and helps us understand how we came to be who we are.
Right.
But what about the future of genetics research? Where do we go from here? What are some of the things on the horizon?
The future of genetics is incredibly exciting. Thanks to advances in technology, especially in DNA sequencing, we can now read the entire human genome, all 3 billion base pairs, with remarkable speed and accuracy. This opens up a whole new world of possibilities for understanding our genetic blueprint and how it influences things like our health, our traits, and even our behavior.
Wow, that's a lot of information. What do we even do with all that data? Sequencing a genome is one thing, but making sense of it all is a whole other challenge.
It's a challenge, but also an amazing opportunity. This wealth of information is already changing the way we approach medicine. It helps us diagnose and treat diseases, develop new therapies, and even understand our evolutionary history. We can now ask and answer questions that we could only dream of before.
So let's get specific. What are some of the most promising areas of research that are being fueled by these advancements?
One area that's creating a lot of buzz is personalized medicine. By analyzing a person's unique genome, we can tailor treatments and prevention strategies just for them. It's a big step away from that one-size-fits-all approach to healthcare and allows us to personalize those interventions.
So it's like having a custom-made healthcare plan based on your own DNA.
Exactly. We can identify individuals who are at a higher risk for certain diseases and step in early to prevent or slow down their onset. We can even predict how they might respond to different medications to make sure they're getting the best possible treatment.
That's incredible. It's like having a crystal ball that can guide us towards better health. What other exciting possibilities are there?
Well, gene therapy is another area with huge potential. It aims to treat those genetic disorders right at the source by replacing faulty genes with healthy ones.
So like a genetic repair crew.
Exactly. It's like going in and fixing those typos in our DNA blueprint.
It's amazing to think that we might one day be able to just correct those errors.
I know it is. And although gene therapy is still in its early stages, it's already showing some remarkable success in treating some rare diseases. And as we learn more about the human genome and improve our gene editing techniques, we can expect even more breakthroughs.
This is all so mind-blowing. It feels like we're just scratching the surface of what's possible. It's such an exciting time to be alive and witness these incredible advancements.
I agree. Genetics is a dynamic field that's constantly evolving. There are new discoveries all the time. It holds such incredible promise for improving human health and understanding those fundamental mysteries of life.
Well, I have to say, this deep dive into inheritance has been a truly eye-opening experience. We started with Mendel's garden, and now we're talking about the cutting edge of genetics research. We explored those intricate mechanisms of heredity and talked about the impact of genetic variations. I feel like I have a whole new appreciation for the beauty and complexity of life's genetic tapestry.
It has been quite the journey.
It has. And it's also given me a sense of optimism. With these incredible advancements, we have the potential to not only understand ourselves better, but to actually alleviate suffering and improve human well-being in some pretty amazing ways.
So now that you have a deeper understanding of how traits are passed down and how our genes shape who we are, what questions does all of this raise for you?
So many. I feel like we've opened up a whole new world for me to explore.
That's what it's all about. And remember, the journey doesn't end here. Keep exploring, keep asking questions, and keep that sense of wonder about genetics. It's a field that affects every aspect of our lives.
Great advice. Thanks for joining us on this genetic adventure. Be sure to tune in next time for another deep dive into the fascinating world of science.